So now we'll summarize lipid metabolism, chapter 14. Most of dietary lipids, 98% in fact, are triacylglycerols. So we're going to call those tags, triacylglycerols, the fats and oils. And we talked about those in our section on lipids. So how does digestion and absorption of lipids work? Well, your salivary enzymes are not going to do much to lipids. The lipids are water insoluble, your salivary enzymes are water soluble, so nothing happens much with the fat in your mouth. That's in contrast to carbohydrates. So the fat goes into your stomach, and your stomach is churning and moving that stuff around. You're not aware of it, but it's just doing that all the time. So that churning action is a little bit like a butter churn. It's going to break up the fats into small globules that are going to float as a layer above the other food components. Doesn't that sound lovely? And I believe this is pronounced chyme. Yeah, okay. Chyme, that's a new word to me. That's that thick, semi-liquid material made up of partially digested food and gastric secretions. That sounds so appetizing. High-fat food takes longer to digest, and you probably are aware of that. There's good things about that. It makes you feel full longer. If you eat a very low-fat meal, especially if it's mostly carbohydrates, your body just processes it very quickly, and then your stomach's empty and you're feeling hungry again. So the fat is very satisfying, and part of the reason is because it helps you to feel full because it digests much slower. So about 10% of those triacylglycerols that you ate are hydrolyzed in the stomach, so just a little bit. In the small intestine, your gallbladder secretes bile. We talked about bile salts. Those help to emulsify those tags, and the hydrolysis continues. But the hydrolysis in your small intestine is usually not complete. Usually what happens is you'll cleave off one or two of the fatty acids, so you'll end up with a monoacylglycerol and some free fatty acids. And these will form into fatty acid micelles. The fatty acid micelles are going to have fatty acids, and they'll have some monoacylglycerols and bile from your gallbladder, and they'll form these little micelles. I don't remember if I put a picture. I didn't. The micelles are small enough that they can be absorbed through the membranes of the cells in your intestine. So what happened is the triacylglycerol had one or two of its fatty acids cut off and got emulsified, and then it went through the cell, into the cell, and now it's going to get put back together. So inside the cell, we're going to reassemble the triacylglycerols. I kind of think of it like, well, let's see, what would be good? Well, maybe, maybe like taking the wheels off a bicycle to get it into your car. Now, the whole bike won't fit. You've got a small car. But if you take the wheels off, you can fit it back in, and then when you get to where you're going, you take it back out and you put it back together. So these, got cut up so that they could get through the cell wall, and then they get put back together. Chylomicrons are lipoproteins that transport these tags from the intestinal cells. So these chylomicrons form from the reassembled triacylglycerols, membrane lipids, and proteins. And what they do is they transport the tags. They go via the lymphatic system eventually to the bloodstream. In the bloodstream, the tags are finally hydrolyzed completely to glycerol and free fatty acids. Those products, the glycerol and fatty acids, are absorbed by the cells, and then they are either broken down to acetyl-CoA to be used as energy, or they may be stored as lipids. If they're stored, they're going to be put back together. So let's go back to this. We looked at the triacylglycerols like this a lot. The glycerol backbone and 3 fatty acids on them. And so they're almost like transformers, you know, and you can take this arm off, and you can take that leg off, and then you can put them back together. And they come apart and get put back together a lot, as needed. So here's an overview for the lipids. Not much happens in your mouth. They get churned up, partially hydrolyzed in your stomach. They go into that chyme as fat droplets. You've got some monoacylglycerols. And then in your small intestine, they get solubilized. And more hydrolyzation occurs. You've got monoacylglycerols and the fatty acids forming the micelles. So these now can get into the cells where the triacylglycerols are reassembled and packaged up as chylomicrons, which can go eventually, well, they go to the lymphatic system first, and then they go to the bloodstream, and then they're rehydrolyzed in the bloodstream to free fatty acids. And then those go off, and they can be either stored or used for energy. Most cells can only store a little bit of fat, of the triacylglycerols. But then there are the fat cells, the adipocytes. And so adipocytes are triacylglycerol storing cells. And most of the cell volume is just a big droplet of triacylglycerol. So here we've got a little cell nucleus over here, but most of the cell is basically an oil droplet. And as more triacylglycerol comes in, it initially forms little tiny drops along the edge, but then they eventually merge in, and you've got one big globule of triacylglycerols. When we talk about adipose tissue, that's areas of your body that contain large numbers of adipocyte, these fat-storing cells. And there are functions for fat in your body, this fatty tissue. It's storage of energy. We'll see that fat gives you a lot more energy for the number of carbon atoms than carbohydrates do. It insulates you from cold, because your body needs to maintain a certain temperature in order for all of your biochemical reactions to occur. And the fat acts as almost like a jacket to keep you warm. And it also protects your organs from physical shock. So we know that there's a lot of adipose tissue, both inside the abdominal cavity and around the abdominal cavity, and that serves to cushion your organs so that if you get hit, you're not as likely to rupture your spleen or something. The term triacylglycerol mobilization, it reminds me of mobilizing the troops. That means the hydrolyzation or the hydrolysis of those triacylglycerols that are stored in the adipose tissue. So you've got all these stored tags. The mobilization gets triggered by hormones saying, hey, we need energy. And so those tags are hydrolyzed and mobilized. Your fat reserves are your body's major source of stored energy. Remember, the glycogen gets used up pretty quickly. The fat takes longer. And so if we look here, this chart's kind of interesting. Stored energy reserves for a 150-pound person. So your blood glucose, only about 80 kilocalories. All your glycogen, 720 kilocalories. That's not going to last you very long. Protein is more, 24,000, but the fats as triacylglycerols, 135,000 kilocalories. That's 84% of the energy that's stored in your body is stored in your fat. Which, if you're not wanting to have as much stored energy, that can be a problem too. So when that is mobilized, when the triacylglycerols are mobilized, they're hydrolyzed and they produce glycerol, because that was the backbone, right? You get the fatty acids and the glycerol. The glycerol is going to travel to the kidneys or the liver, and it gets converted to dihydroxyacetone in a two-step process. Dihydroxyacetone is one of the intermediates in both glycolysis and gluconeogenesis. And so it can be converted to pyruvate, or it can be used to form glucose. So whichever way is needed. And that is an example of how lipid and carbohydrate metabolism are connected to each other. So that's the glycerol. Then the fatty acid, what happens to them? Well, there's three parts to the process in which they are broken down. There's activation. So they bind to coenzyme A to form acyl, not acetyl, acyl-CoA. And the difference there is acetyl-CoA is 2 carbons, acyl-CoA is any length of carbon chain. And we learned that the fatty acids, they could be C16, C18, C20, they vary in length. Then that activated fatty acid is transported to the mitochondrial matrix by a shuttle mechanism, and there it undergoes oxidation. In the oxidation, we have 2 carbon units cut off from the carboxyl end of the acyl-CoA. So it's like you got this big, I don't know, big roast or something all ready to go, and you put it in this place, and then it slices stuff off the end. That's how I'm picturing it in my head. And that oxidation is called the beta oxidation pathway. And that is just, it's a repetitive series of 4 reactions that takes the acyl-CoA down to acetyl-CoA by removing 2 carbons at a time. And so this is the process for saturated fatty acids. Remember, saturated fatty acids only have single bonds. They don't have any double bonds. So the fatty acid is originally an alkane attached to the CoA. And so there's dehydrogenation to form an alkene. It's hydrated to form a secondary alcohol. It's dehydrogenated to form a ketone. And then thiolysis is where it's cleaved from the CoA. The CoA bonds to it by a thioether connection. And then for the unsaturated fatty acids, there's an extra 2 steps that we're just not even going to talk about. But you got to do something about those double bonds, because the double bonds are not going to fit into this pathway. So you got to take care of the double bonds before it can fit in here. So here's a summary. So if we're looking at C18, that's stearic acid, I believe. So saturated, so here's 3 and 14 and the rest of these. So we've got 18 carbons here. There's the CoA and the thioether linkage. I guess it's actually a thioester because of this guy. Thioester linkage. And so originally we have dehydrogenation. So look at this orange guy. We're taking hydrogens off of these 2 carbons to make a double bond. Then we're adding water to make an alcohol. Then we take the hydrogen off and make this ketone. Now this part is looking kind of like that part, isn't it? And then this part gets cut off. And the CoA attaches, another CoA comes in and attaches here. So now what's happened is we've lost 2 carbons. We went from 18 to 16. And then it just does this over and over and over and over again until it gets down to the end where you're breaking this off. You get 1 acetyl-CoA here and another acetyl-CoA there, and you're done. So the product of this beta oxidation is acetyl-CoA, which is this guy right here. Did you notice when we talked about fatty acids that they're all even numbers? What would happen to this process if they weren't even numbers? We wouldn't end up with acetyl-CoA at the end. We'd end up with something with 3 carbons or 1 carbon. It wouldn't work very well. And when these fatty acids are formed, they're formed in a similar process where 2 carbons are put on at a time. And so all of these fatty acids are going to be an even number of carbon atoms. So let's compare energy production. So for that stearic acid, the C18, here they're just tallying up all the different individual reactions that we didn't look at. The gross here is 122 ATPs from one stearic acid molecule. There are 2 ATPs required for the initial activation, however, so you have to subtract those off. And the net production is 120. Do you remember how many we got from one glucose molecule? 30. Now, the stearic acid is larger, right? It's 18 carbons instead of 3 carbons. But the difference is more than that. And so if you compare them based on equal numbers of carbons, the lipids are 33% more efficient as energy storage than the glucose, the carbohydrate is. I think that's kind of interesting.
I have a question.
Mm-hmm.
So we get a lot of energy from fat, right?
Yes.
And then when we eat, we get energy from calories?
Well, calories come from fat, protein, and carbohydrates.
Okay, and then what's in caffeine? Because that kind of wakes us up.
Caffeine wakes us up. Caffeine stimulates our central nervous system. And so it causes our body to react to things a little differently. And so I'm not sure exactly the mechanism of it.
It binds to a receptor and it causes a longer response than something like, for instance, like acetylcholine.
Okay. So like the epinephrine we saw.
So it's kind of like adrenaline or something?
Yeah, a little bit,
Okay.
but it lasts longer. So adrenaline causes you to bring more glucose from your glycogen stores to provide immediate energy. And so something like caffeine, if that acts in the same way, it's going to cause the same thing to happen. And, you know, so everything that we eat has an effect on us. And, yeah, I think there's just, the body is very, very complicated. And even expert medical doctors and researchers, nobody really understands how everything works. And that's why we've got so many conflicting theories. Of course, one of the problems is that it's hard to do real experiments on humans because they don't like being cut up afterwards, right? They don't like being messed with. And so we just have to observe and do very limited experiments. Well, that's a good question. Here's some generalizations about how your body uses fuel, food. I thought this was pretty interesting. So your skeletal muscle uses glucose from the glycogen when it's in an active state. But in a resting state, it uses fatty acids, which would come from lipid metabolism. Your cardiac muscle, your heart, depends first on fatty acids and secondarily on ketone bodies, which we'll talk about in a minute, glucose, and lactate. Your liver prefers fatty acids. Your brain needs glucose. It can also use ketone bodies. Your brain can't use fats because they can't cross the blood-brain barrier. Now, your body can do things with the lipids and make them into glucose that can get to your brain, but it can't go there directly. So I guess if people call you a fathead, you could say, well, technically, I can't be a fathead because fat can't cross my blood-brain barrier. And that would put them in their place, wouldn't it? Ketone bodies. So we just looked at the beta oxidation of fatty acids and how it forms acetyl-CoA as the product. This acetyl-CoA is further processed through the citric acid cycle, which we studied last week. That was in chapter 12 about the overview of metabolism. Well, the citric acid cycle is not going to function sometimes. We can't use the acetyl-CoA in the citric acid cycle if we've got a lipid-carbohydrate imbalance. And this can happen for one of 3 reasons. If your dietary intake is high in fat and low in carbohydrates, the acetyl-CoA is not going to be able to be used in the citric acid cycle. If you have a diabetic condition where the glucose is not being used like it is supposed to be, you can have this happen. Or under prolonged fasting conditions, and that would include starvation. And under these conditions, the acetyl-CoA is still used, but now it's diverted to formation of ketone bodies. So what are ketone bodies? Well, these are the 3 ketone bodies. There's acetone, beta-hydroxybutyrate, and acetoacetate. These are substances produced when you've got extra acetyl-CoA that's accumulating because your body can't send it to the citric acid cycle because it's got a lipid-carbohydrate imbalance. And ketone bodies were originally thought to be just sort of degradation products and not actually beneficial. But now we know that they actually serve as energy sources. They're especially important as energy sources for heart muscle and the renal cortex. Those parts of your body can use ketone bodies very well. And if you're on a low-carb diet where your blood sugar is lower and you're not getting a lot of glucose into your brain, your body can use ketone bodies as fuel for your brain. And so personally, I've been interested in low-carb diets. And it first came up the first time I was teaching this class, and I saw a video about it, and it's like, you know, biochemically, that makes so much sense. And of course, you know, people say, oh, well, ketosis, and it's all bad, and this and that and the other thing. But the more I look into it, the more sense it makes. That you don't have to have a large carb intake to be healthy. Ketogenesis is the process by which ketone bodies are synthesized. So keto should make us think of ketone bodies, genesis, making. Ketogenesis, making of ketone bodies. So again, this is a multi-step process. Here we got 2 acetyl-CoA molecules, and they're going to undergo condensation. Here's another acetyl-CoA coming in, and another condensation. Remember, a condensation reaction is where you have 2 molecules combining. And so we've taken 3, see this is a C2 and a C2. We combine them, we get a C4. We add in another C2. Now we've got a C6. Then that gets cleaved. And now we're back to a C4. We've got acetoacetate. That's one of the ketone bodies. And we can have hydrogenation and get beta-hydroxybutyrate is one of the other ketone bodies. So the word ketosis just refers to an overall accumulation of ketone bodies in the blood or urine. And that means that your body, instead of sending acetyl-CoA to the citric acid cycle, is converting it to ketone bodies. And generally, this is not harmful. If you are in ketosis, sometimes you can tell because your saliva has kind of a funky smell to it. It kind of smells like acetone because there's actually acetone in your breath. But it's generally not harmful. In fact, those keto sticks that we used in lab last week, those are to measure ketones in your urine. The one exception to ketosis not being harmful is ketoacidosis, which happens or can happen with uncontrolled type 1 diabetes. And what happens here, you've got ketosis going on. 2 of the ketone bodies that can be made are acids, and these can alter the pH of the blood. And when that happens, you've got a serious problem. But this will not happen in someone who is not diabetic. Ketoacidosis can result in coma and death, so that can be pretty serious. So we talked about breaking down lipids. We can also make lipids, so lipogenesis, metabolic pathway in which we synthesize fatty acids from acetyl-CoA. And again, this is not simply a reversal of the beta oxidation pathway. So this is, here we have illustrated a one cycle. So we need these activated components here. These both came from acetyl-CoA, and I decided not to go into the reactions that form those. But they undergo condensation, a hydrogenation, dehydration, and another hydrogenation. The process here of one cycle adds 2 carbons. We started out with C2, and we end up with C4. And every time it goes through this cycle, it adds 2 carbons. And that's why all these fatty acids are an even number of carbons. It's due to how they are made. They are made 2 carbons at a time. And so this is kind of the overview of carbohydrate and lipid metabolism. So carbohydrates undergo digestion to form glucose, and they can go through glycolysis to form pyruvate. The pyruvate can form acetyl-CoA and get into the citric acid cycle. Remember, the citric acid cycle provides electrons for the electron transport chain and generates ATP. Fats get digested. Glycerol is one part, that's the backbone of the lipid. And so that can come over here and enter into glycolysis, or it can be used to form glucose. The fatty acids can be stored, or they can be, they're shuttled into the cell, and, the mitochondria. And then they undergo that beta oxidation pathway. This is a spiral because it's showing you multiple turns. Every turn of it, every cycle, you cut off 2 carbons, you make 1 acetyl-CoA. And that acetyl-CoA generally goes into the citric acid cycle. And so this guy's like this little, like a hamster in a wheel, just going around and around and around. But if you don't have enough pyruvate coming in here to take care of the acetyl-CoA, then it will undergo ketogenesis and form ketone bodies, and those will go out and be utilized for energy. You can also, from this point, from the acetyl-CoA, you can also make fatty acids out of that. So it's very complicated, and yet it's kind of simple too. So have fun with that.